As Paul Levinson once said, and I quote, "Online learning can be a lifeline to those who have obstacles, such as geographical distances or physical disabilities." And what better way to use this platform in these times of corona uncertainty? Well, welcome everybody. I'm Dr. Sanskruti Patel, and in this lecture, you'll learn about the interesting concepts of pharmacodynamics. This will be the outline of pharmacodynamics, and today we will mainly focus on the site and the mechanism of drug action. But before that, let's revise the pharmacokinetic concepts. You already have studied this, so let's just quickly revise. The drug may enter into the body by a variety of ways, as an oral liquid, pill or capsule, as an inhaled vapor or aerosol. It can be absorbed through intact skin or mucous membrane. It can be injected into muscle, subcutaneous tissue, spinal fluid, or directly into the bloodstream. As we shall see, the physical properties of the drug and the specific way it is prepared, it greatly influences the speed of absorption. Now, if the drug is given orally and swallowed, it must be absorbed from the GI tract into the portal circulation. And if it is absorbed through the skin, mouth, lungs, or muscle, it will go directly into the systemic circulation. If the drug is injected directly into the bloodstream, that is, for example, intravenous injection, 100% of it is available for the distribution to the tissues, and this is not usually the case for other modes of administration. For example, a drug which is absorbed via the portal circulation must first pass through the liver, which is the primary site of drug metabolism, that is known as biotransformation. Some of the drug may therefore be metabolized before it ever reaches to the systemic blood, and in this case, the first pass metabolism reduces the bioavailability to less than 100%. I'm sure you're quite familiar with all these terms, first pass metabolism and bioavailability by now. So now once the drug is in the bloodstream, a portion of it may exist as free drug dissolved in the plasma water. Some drug will be reversibly taken up by the red cells and some will be reversibly bound to the plasma proteins. For many drugs, the bound forms can account for about 95 to 98% of the total, and this is very important because it is the free drug which traverses the cell membrane and produces the effect, right? So it is also important because the protein bound drug can act as a reservoir which releases drug slowly and thus prolongs its action. Now the unbound drug, so what happens to that? So the unbound drug may then follow its concentration gradient and distribute to the peripheral tissues. Now in some cases the tissue contains the target site, and in other the tissue is not affected by the drug. And these sites of non-specific binding, they act as further reservoir for the drug, right? So the total volume of distribution determines the equilibrium concentration of the drug after a specified dose. Now what happens to these tissue-bound drugs? So they eventually re-enters the bloodstream where it perfuses the liver and kidneys, and the liver metabolizes most drugs into inactive or less active compounds, you can say, which are more readily excreted. Now these metabolites and some of the parent compound may be excreted into the bile or may pass out of the body via the feces, right? Or alternatively, some of the drug may be reabsorbed again and further down into the gastrointestinal tract, which is so-called enterohepatic cycle. And any biotransformed drug which is not excreted in the bile, it passes back into the systemic circulation. So now the parent drug and the metabolites in the bloodstream, they may then be excreted, and most of them are filtered by the kidney, where a proportion undergoes reabsorption and the remaining is excreted into the urine. Now there are some drugs which are actively secreted into the renal tubule, and another route of excretion can be lungs, like drugs like alcohol or anesthetic gases. They are eliminated via lungs. and also smaller amounts of smaller amount of drugs are eliminated in the sweat, tears, and breast milk. So this concludes our overview of the pharmacokinetics. So the question arises that what is pharmacodynamics, right? So it is basically a Greek word, pharmakon, which means drug, and dynamics, which means action or activity, right? So pharmacodynamics, it is a study of biochemical and physiological effects of the drugs and their mode of action. It deals with the relationship between the plasma concentration of drug and its response and duration of action. Or in simpler words, you can say it is what a drug does to the body. So what do you mean by drug action and drug effects? Well, they are often used as synonyms. Like the drug action, it is the initial interaction of the drug with the cells at the site of action, and the resultant physiological and biochemical consequences are its drug effects, right? So to understand this, let's take an example of one drug, right? So pilocarpine, say for example. So pilocarpine, it is a parasympathomimetic drug, which is also known as cholinergic agonist, right? So when this drug is given to the patient, it will cause the parasympathetic stimulation of the circular muscle of the iris. So this will be your drug action, right? And the meiosis that is produced is the drug effect. So meiosis is the constriction of the pupil of eye, right? Also always remember that the drug action precedes the drug effect, and these two, they depend on the drug concentration which is achieved at the site of action, which is determined by these points. The first will be your absorption of the drug, then first pass metabolism, distribution, biotransformation, excretion, tissue affinity. For example, there are ultra short acting barbiturates, for example, sodium thiopental, which is a highly lipid soluble drug, and it mainly gets concentrated in the central nervous system. Then we have the condition of the body or the milieu interior, which means that so say for example, if the person is having iron deficiency anemia, then the iron will be absorbed more rapidly in such individuals, right? So prior to understanding this, we must know that where the drugs can act and their site of action. So the drugs may act at three sites. The first is the extracellular site of action. For example, antacids, they are the drugs that helps to prevent or correct or neutralize the acidity, right? Then we have chelating agents that forms complexes with the heavy metals like lead, copper, mercury, etc. Then the next example is of magnesium sulfate that acts as an osmotic purgative. Like, see, for example, if you give a patient a magnesium sulfate, what it will do, it will retain the fluid inside the lumen of the intestine, and it will increase the fecal bulk, and therefore it is used to treat the patients having constipation, right? Then we have the cellular site of action. So there are receptors which are located on the cell membrane, and the ligands or the drugs, they bind onto these receptors and then they produce the effect. For example, acetylcholine, it acts on the nicotinic receptors of the motor end plate and causes the contraction of skeletal muscle. In other examples, we have the inhibition of membrane-bound ATPase by cardiac glycoside drugs and the effect of sympathomimetics on the heart muscle and blood vessels. Lastly, we have drugs that act on intracellular site, and for this we have a wonderful example of trimethoprim, or you can say sulfa drugs. So what they do is they interfere with the folic acid synthesis, which is an intracellular component, right? And of course, the example of 5-fluorouracil, which is an anti-cancer drug, it gets incorporated into the messenger RNA in the place of uracil. So now moving on to the mechanism of action of drugs. The majority of drugs, they act by receptor mediated mechanism, or by non-receptor mediated mechanism, or by targeting the specific genetic changes. So first, let's learn about the non-receptor mediated mechanisms. So a drug produces a quantitative and a qualitative change in the function of the target organ. So they may produce their effects by the following ways. So the first one is stimulation. So any increase in the activity of specialized cells is called as stimulation. For example, adrenaline stimulates the heart. Pilocarpine, when given, it stimulates the salivary glands and thus salivary gland secretion. Now also keep one thing in mind, that excessive stimulation is followed by depression of that particular function. For example, picrotoxin, it is a CNS stimulant, antidote, convulsant, and GABA antagonist, right? It can cause coma and can ultimately lead to death. The other mechanism is depression. So depression, it is the decrease in the activity of the specialized cells. For example, barbiturates, they depress the CNS. Quinidine depresses the heart. Acetylcholine depresses the sinoatrial node. Also, it stimulates the smooth muscle tissue. Then we have irritation. So irritation, it indicates that drug produces adverse effect on the growth, nutrition, and morphology of the living tissues. For example, senna and cascara, right? So what they do is they are mild irritants. So they cause mild irritation and they stimulate the mucosal cells of the gut, and it acts as laxatives, right? Then we have counter irritants, which are applied to the skin, which is situated over the organ which is responsible for the pain. The next entity is replacement. Like for example, in an iron deficiency disease or condition, the iron stores are depleted. So we need to replace or replenish them, right, by providing the iron supplements. Similarly, in Parkinson's disease, the dopamine levels needs to be elevated, right? You need to give externally dopamine by giving the patient the combination of levodopa and carbidopa. And in cytotoxic action, we have these wonderful examples of penicillin and chloroquine. Now, these are the non-receptor mediated mechanisms, which can also be classified as follows, like by chemical action. We have already studied these, so we'll just go quickly. So first is your chemical action, in which we have neutralization, right? We have already studied about the antacids. They act by neutralizing the gastric hyperacidity. Then we have the anticoagulant action of heparin, right? So heparin, it is a strongly acidic mucopolysaccharide which acts by neutralizing the basic group of various clotting factors and thus prevents the action of thrombin, right? Then we have chelation. So there are various chelating agents which trap these heavy metals and form the complexes, and they are finally excreted out of the body. They are mainly used to treat heavy metal poisoning. For example, EDTA, which chelates calcium. Then we have calcium disodium edetate, which chelates the lead. Then we have dimercaprol, which chelates the mercury. Then we have penicillamine, which chelates the copper, and deferoxamine, which chelates the iron, right? So all these drugs are used to treat heavy metal poisoning. And lastly, we have ion exchangers, right? So anion exchange resin, that is cholestyramine, what it does is it exchanges the chloride ions from the bile salts, and the resultant complex, it is not absorbed and it is excreted out, and therefore this drug is used as a cholesterol lowering agent. Then by physical action, so first we have already studied this magnesium sulfate. It acts as an osmotic purgative, and next is adsorption. So for adsorption we have kaolin, which absorbs the bacterial toxin and it acts as an antidiarrheal agent. Then we have methyl polysiloxane and simeticone, which absorb the gases and they act as anti-flatulent, right? Then we have various dusting powders. They act as protectives to provide the local effect. Then we have demulsants. Now demulsants are the drugs that coat the inflamed mucous membrane and they provide a very soothing effect, right? For example, pectin. So it is basically used in antidiarrheal preparation. Then we have menthol and ceravasica, which is used in cough phlenictis, right? The next entity that we have that acts via physical action is astringents. So what they do is they precipitate and denature the mucosal proteins and they protect the mucosa by firming up the mucosal surface, right? And example is tannic acid in gum paints. And then we have saturation in the biophase. So what does it mean? For example, general anesthetics, right? So they simply saturate these general anesthetic agents. They saturate the cellular site of the central nervous system and they get arranged in between the membrane lipids and they thus hinder some metabolic functions or disrupt the membrane organization, and this is how they act. Then the third type is by counterfeit or false incorporation mechanism. We have already studied this. Example was trimethoprim 5-fluorouracil. Then we have other sulfa drugs, right? So they basically interfere with the bacterial synthesis of folic acid from para-aminobenzoic acid, and therefore it prevents the growth and development of that bacteria. Then we have by virtue of being a protoplasmic poison. So there are certain drugs like germicides and antiseptics like formaldehyde and phenol. They act non-specifically as protoplasmic poisons, which causes bacteria death, right? Then through the formation of antibodies, so there are various vaccines that produce the effect by inducing the formation of antibodies and it stimulates the defense mechanism of the body. For example, vaccines which were made against smallpox and cholera, they provide active immunity, and the antisera, which is used against tetanus and diphtheria, it provides passive immunity, right? Then through the placebo action. Now, what do you mean by placebo? So placebo is basically a pharmacologically inert and harmless substance which resembles the actual medicament, which is exactly same in size, in shape, in color, in smell, and in weight, right? And the examples of placebo that are used in solid dosage forms are starch and lactose, right? So the patient that reacts to such placebo treatment are called as placebo reactor, and the placebo effect is not at all mediated through any receptor action. It is simply the faith in the treating physician which the patient has, right? And lastly, but not the least definitely, we have the action which is by targeting the specific genetic changes. So example is of course anti-cancer agents that specifically target the genetic changes, the inhibitors of RAS modifying enzymes and the inhibitors of specific tyrosine kinase that block the activity of these oncogenic kinases. So now moving forward to the receptor mediated mechanisms. Right? So majority of the drug, they produce their effects through an interaction with the chemical component of the living cell. This chemical component is called as receptor. So a receptor is a specific macromolecular protein which can be membrane bound or can be located intracellularly, which is capable of binding with specific functional groups of the drug or endogenous substances. Now, as you can see here, the equation given, binding of a drug with its receptor, it results in the formation of DR, that is your drug receptor complex, which is responsible for triggering the biological response. Now, this binding is usually specific and reversible. Reversible only when there is formation of hydrogen bonds or van der Waals bonds or electrostatic bonds. But this binding is irreversible when there is formation of covalent bond, right? So the overall drug effect is attributed to the following two factors. So the first is your affinity. So what is affinity? So affinity is the capability of the drug to form a complex with its receptor, that is the DR complex. For example, you have a key. So key entering the keyhole of the lock, it has got affinity to its levers, right? Then we have intrinsic activity or efficacy. So intrinsic activity, it means the ability of a drug to trigger the pharmacological response after making the drug receptor complex. For example, if the same key that I had earlier, after entering into the keyhole of the lock, if it can open the lock too, it has got an increasing intrinsic activity, right? And if it cannot open the lock, it is called as the key or the molecule only has the affinity for that lock or for that receptor, right? So on the basis of affinity and efficacy, the drug can be broadly classified as agonist, antagonist, and they are further subclassified as partial agonist and inverse agonist, right? So let's discuss it one by one. So the first, that is agonist, right? So these drugs or agonists which bind to the receptor, they can trigger the maximal biological response, or it can mimic the effect of the endogenous substance after combining with the receptor. So the affinity and the intrinsic activity for agonist are both high, right? Then we have antagonist. So these drugs, they bind to the receptors, but they do not mimic. They rather block or interfere with the binding of an agonist, right? So antagonist, definitely they have the affinity, but they lack intrinsic activity. Now the question arises that when two drugs are binding to the same receptor at the same site, why is it that one drug is acting as an agonist and the another drug is serving as an antagonist, right? So for this, to answer this, you have to first understand the concept of dual nature of receptors. Now also remember that for any drug to produce response, it has to bind to the receptor, and not only that, it must produce a conformational change in the structure of the receptor so that the response will be manifested, right? Just keep this thing in mind. Okay, so with this, let's move ahead. So receptors exist in at least two conformations. The first is the active, that is represented as RA, and inactive, that is RI, right? So it exists in two conformations, active and inactive. So normally RA and RI exist in equilibrium, and the relative affinity of the drug for these two conformations will decide whether the drug will be an agonist or an antagonist. Okay, I repeat this again. So the relative affinity of the drug for these two conformations will decide whether the drug will act as an agonist or as an antagonist when it binds to the receptor. For example, there are two scenarios. So first one is when the drug has high affinity. Okay, so when the drug has high affinity for active conformation RA, then for the inactive, that is RI, such a drug will act as an agonist and it will shift the equilibrium towards the active state, right? And it will activate the receptor and the response will be obtained. Whereas the second scenario, when the drug binds to both of these conformations, that is when drug binds to both Ra, that is active, and Ri, that is inactive conformation with equal affinity, right? Such drug will serve as an antagonist, as it will settle the equilibrium. So what I mean is, it would neither activate the receptor nor will it shift the equilibrium towards any site. So there will be no response. There will be affinity, but there will be no activity, no intrinsic activity, right? So now partial agonist. So they have the full affinity to the receptors, but they have low intrinsic activity. For example, pentazocine, which is a narcotic analgesic, it is a partial agonist at the mu receptor, which is a subtype of opioid receptor, right? They have a slightly higher affinity for the active conformational form Ra than the inactive form, right? And thus they display an intermediate effectiveness between agonist and antagonist, right? Now we have inverse agonist, right? So these have the full affinity towards the receptor, but the intrinsic activity can range from 0 to -1. That is, it produces effect, which is just opposite to that of agonist, right? For example, beta-carbolin, which is an inverse agonist for benzodiazepine receptors, right? So it produces effects like anxiety, insomnia, and seizures, which are just opposite to the therapeutic effects of benzodiazepine, which is anti-anxiety, sedation, and anti-epileptic. So this slide is just the recap of what we learned, and to summarize the two-state receptor model, you know that receptors have two essential functions, which is the recognition of the specific ligand molecule and transduction of the signal into response, right? Also, on these receptors, we have two domains. The first one is ligand binding domain, which is your coupling proteins. And the second is your effector domain, which undergoes the functional conformational change after the drug binds to the receptor. Okay, so this conformational change then results into two things: action and effect, right? So in action, initially there will be combination of the drug with the receptors that results into the conformational change, and in effect it is the ultimate change in the biological function which is brought about as a consequence of the drug action through a series of intermediate steps, that is transducers. So this transducer mechanism, it is basically a very complex multi-step process which involves the amplification and integration of the signals which are received in the form of extracellular or intracellular signals, right? So there are large number of receptors which share these handful of transducer mechanism which generate an integrated and amplified response. And there are four major categories of these. So the first one is the GPCR, that is your G protein coupled receptors. Then we have receptors with intrinsic ion channel. Then we have enzyme linked receptors. And lastly, we have the transcription factors, that is the receptors for gene expression. So first, let's start with the GPCR, that is G protein coupled receptors, or also known as metabotropic receptors. So these are the membrane bound receptors which are coupled to the effector system through GDP, that is guanosine diphosphate, or GTP, that is your guanosine triphosphate binding proteins, which are called as G proteins. So these G proteins, they include muscarinic cholinergic receptors, adrenoceptors, dopaminergic receptors, 5-HT receptors, opiate receptors, and purine receptors. I know this will be a bit difficult for you guys, but as you will learn further in pharmacology, you will be able to correlate things when you will study again the general pharmacology. So as of now, you just have to accept, and you just have to mug it up, right? So these were the examples of GPCR. And when we talk about the GPCR structure, GPCR are integral membrane proteins that possess the 7 membrane spanning domains or transmembrane helices. And these G proteins are heterotrimeric molecules, that is, they have 3 subunits, which are alpha, beta, and gamma. And these 3 subunits, alpha, beta, and gamma, are so tightly bound together that they did not dissociate from each other, and therefore they are written as a single thing, right? Alpha, beta, gamma subunit, right, as a single thing. Also, in a single transmembrane domain, it has 2 parts, the extracellular domain and the intracellular domain. So the extracellular domain, that is, let me show it to you. That is this extracellular domain. It has the ligand binding site. So this will be your ligand, that is your agonist in this case. This is your ligand binding site. And then you have the intracellular domain, which is responsible for the G protein based signaling modulation. Now, you can see the slide shows varieties of G protein receptors, G proteins in fact. So as I mentioned earlier, there are 3 subunits, alpha, beta, and gamma, right? Their further classification is based on the identity of their distinct alpha subunit. So it is believed that there are 3 main varieties of G alpha proteins, that is GS, GI, GQ, right? Although there are other 17 variants which are known to us. So among these, the GS and GI. So GS, S stands for stimulation. So GS stimulates the adenyl cyclase, whereas GI, I stands for inhibition. So GI inhibits the adenyl cyclase. Whereas GQ, it stimulates the phospholipase C activity. Okay. So this slide shows us the mechanism of activation of G protein. Now it is very interesting and very easy. So you can see here in the resting stage, okay. So you can see that in this resting stage, the alpha, beta, and gamma subunit of G protein is linked to one another, and of course GDP. Okay, GDP is also bound to the alpha unit, and binding of an agonist, say for example in this case, if we take epinephrine, if it binds to a G protein coupled receptors, that is, if this is your beta 1 adrenoceptor, it causes exchange of what it causes? It causes exchange of GTP for GDP on the alpha subunit, right? And the beta and the gamma, that is your beta and the gamma subunit, it dissociates from the alpha and the GTP complex, which now interacts with the target protein. So you can see as soon as the agonist binds to the receptor, the alpha and the GTP, which is converted from GDP, will dissociate from the rest of the subunit, that is your beta and gamma subunit, right? So this will activate it, and the activated effector, it then propagates the transduction mechanism through a set of secondary messengers, right? So the first messenger was the agonist, as we see, right? The agonist came and it binds to the receptor. It will be your first messenger. It will be your first step where you will encounter the second messenger, and then when the agonist is no longer present at the receptor, reverts to its resting stage, right? So in this process, the GTP on the alpha subunit, it will be hydrolyzed to GDP again, and the effector protein, that is adenyl cyclase, will be inactivated, right? So this is a cycle. So this slide shows the GPCR signal transduction, and there are three G protein coupled effector system. The first one is your adenylate cyclase, that is your CAMP system. The second is your phospholipase C inositol phosphate system, and the third is your ion channel regulation. So starting with the adenylate cyclase, that is your CAMP system. You all are aware that CAMP, that is cyclic AMP, is a second messenger and is synthesized by adenyl cyclase under the influence of different G protein coupled receptors, right? And you are also familiar with the fact that GS will stimulate the adenyl cyclase, GI will inhibit the adenyl cyclase, therefore it will reduce the CAMP, right? So this figure on the slide, it shows an example the ways in which there will be increased CAMP production in response to the beta adrenergic activation. So this receptor present is your beta adrenergic receptors, and how it is activated and how the calcium will be released and how the system works. Okay, so let's see. So what happens is as soon as this norepinephrine comes and it binds to the beta adrenergic receptors, the G protein will be activated, which will further cause the adenyl cyclase activation or stimulation, and it will help in the conversion of inactive protein kinase to active protein kinase, right? And because of which there will be the entry or the influx of the calcium ions. Now the calcium ions, as soon as they'll get into the muscle or the tissue, it will cause, it will act on the sarcoplasmic reticulum, right? It will further release the calcium, which will bind to the troponin, and there will be actin myosin interaction seen, right? And this will cause increased cardiac contractility and increased cardiac velocity, right? So in short, phosphorylation of these channel increases the calcium entry into the cell during the action potential, which will lead to increase in the force of contraction of the heart muscle. So this will be our final action that we will get, right? Also, adenyl cyclase or CMP system, it is involved in energy metabolism, cell division and cell differentiation, ion transport and ion channels, and of course contractile proteins which are present in the smooth muscle. Now this was all about how the system works and how it is activated and how the response we get, right? But how the adenylase cyclase system will be inhibited? So as I said, GI will be stimulated, which will cause inhibition of adenyl cyclase. It will cause decreased CMP production, right? And examples of these are M2 receptors, which are muscarinic cholinergic M2 of cardiac muscle. Then there will be alpha 2 adrenoceptors in smooth muscles and opioid receptors. Okay. Also keep one thing in mind, that the cyclic AMP will be hydrolyzed within the cells by PDE, that is phosphodiesterase enzyme, to 5-AMP, and this is how its action will be dominated. Okay, always remember, if agonist comes and binds to the receptor, it will produce this effect of increased cardiac contractility, but we don't need the stimulation all the time. So to keep things in check, we also need the inhibition system, and this is how the functions are regulated. This slide shows us the phospholipase C inositol phosphate system. Now it is very interesting and very easy. So there are some hormones, right? There are hormones in the body, of course, like vasopressin, TSH, that is your thyrotropin releasing hormone. Then you have neurotransmitters, various neurotransmitters and growth factors. So all these, they bind to the receptors which are linked to the G protein, that is your GQ, right? and they activate a membrane enzyme called as phospholipase C. Now this phospholipase C stimulation, it will lead to the hydrolysis of PIP2, that is phosphatidyl inositol 4,5-diphosphate. Okay, so the hydrolysis of PIP2 will give us two products, DAG and IP3. So DAG is your diacylglycerol and IP3 is your inositol 1,4,5-triphosphate. Now as you can see, DAG will remain confined to the membrane, while IP3, your IP3 is water soluble and it can easily diffuse into the cytoplasm, where it will trigger the calcium release from the storage vesicles, and this will lead to the calcium, exactly, this will lead to the calcium release, and increased concentration will promote the binding of calcium to calmodulin, right? So calmodulin and calcium, they will cause, it will, what it will do, it will produce various cellular response. Okay, also there is DAG, as we have discussed, which remains confined to the cell membrane. Now this DAG, it activates a phospholipid and a calcium sensitive protein kinase C, right, which then phosphorylates the specific protein substrate, leading to the response. Okay, so this is the normal thing. So the important key points to remember is PIP2 is the substrate of the membrane bound enzyme, okay, which splits into two, DAG, PIP2. DAG remains confined to the cell membrane and later it activates the protein kinase C, right? Then we have IP3, it will stimulate the release of the calcium from the storage vesicle, will cause the binding of calcium and calmodulin, and it will produce the cellular response. Then the next thing is the ion channel regulation, right? So G protein coupled receptors, they control the functioning of ion channels like potassium, sodium, calcium channels by mechanisms that absolutely do not involve the second messengers, right? So what are second messengers? For example, your CAMP or your inositol phosphate, right? So to understand it better, let me give you an example. In cardiac muscle, we have muscarinic acetylcholine receptors, which are known as M2 receptors, right? So these receptors, they are known to enhance the K2, that is your potassium permeability, that is thus hyperpolarizing the cells, and it inhibits the electrical activity, right? The second example will be your opioid analgesics, which reduce the neuronal excitability by opening these potassium channels, right? Now moving on to the receptors with intrinsic ion channels. So ion channel receptors, they are also known as ionotropic receptors, right? So these are the receptors that are located on the cell membrane. They are coupled directly to an ion channel and therefore they are known as agonist regulated ion channels. So as the name suggests, agonist gated ion channels or agonist regulated ion channels, they only open when the receptor is occupied by an agonist. As you can see here, only if the receptor is occupied by agonist, only then it will open the ion channels, right? And then there will be subsequent flow of the ions through these channels, which will cause or which will produce the cellular response in the form of depolarization or hyperpolarization of the cell membrane, right? Now the ligand gated ion channels are also known as ROCs, that is your receptor operated channels. Okay, so the next entity in this is your voltage operated ion channels. So in voltage operated ion channels, the ion conductance or the ion transfer is modulated through alteration in the voltage gradient across the plasma membrane, right? For example, you have nicotinic cholinergic receptors, then GABA A receptor, then glutamate receptor, glycine receptor, and some of the drugs are the allosteric modifiers of channel gating. For example, benzodiazepine, which allosterically enhance the chloride transport through the GABA A chloride channel. As of now, you have to mug these examples and there is no way out. But as you will progress into the subject of pharmacology, everything will make sense. So as of now, you just have to, what can I say, tolerate it. You have to mug it up. Okay, so this is the example of ion channel coupled receptors. That is your nicotinic acetylcholine receptors, which is operated channel. It consists of 5 proteins. 5 protein subunits to be specific. There are 2 alpha, as you can see in the diagram. There are 2 alpha, 1 beta, gamma and theta, right? So all of them, they pass across the cell membrane and they are arranged in such a manner that there is a central pore, and in order to activate the receptor and open the channel, one molecule of acetylcholine should bind to each of the alpha subunit. As you can see here, acetylcholine needs to be here on the alpha subunit. They needs to bind on the alpha subunit, and then the ROC, that is your receptor operated channels, becomes an open state. Okay, and this will open up the conductance of ions, and this entire transduction is for a very few seconds. You can say for milliseconds. It is very rapid, right? So the next is the enzyme linked receptors. Okay, so basically enzyme linked receptors, they are a group of transmembrane proteins that contain either intrinsic enzyme activity on their intracellular domain, or they are associated directly with an intracellular enzyme. Now, upon the ligand binding, it causes a conformational change which is transmitted via a transmembrane helix. As you can see in this diagram, it is transmitted via a transmembrane helix which activates the enzymes and initiates the signaling cascade, right? Also, there are basic receptor structures you are already aware of. In the extracellular part, we have ligand binding domain, which is easily accessible for the ligands. They have the strong affinity for specific ligands, and it allows different ligands to bind to the same receptors in order to evoke particular cellular response. Then we have a transmembrane domain which contains a series of hydrophobic amino acid to to tether the receptor to the cell membrane. And the third is the cytosolic or the active enzyme domain. So first was your extracellular, then it was transmembrane, and then we have the cytosolic or active enzyme domain, which is basically the intracellular domain of receptor, which itself is an enzyme and interacts directly with an enzyme, right? So having said that, the basic structure of enzyme receptors, you must also know about different types of these enzyme receptors. So they are classified into, you can say, six varieties of them. The first one is the receptor tyrosine kinase, which phosphorylate the specific tyrosine residues on a small set of intracellular signaling molecules. The second type is of tyrosine kinase associated receptors, where it forms a couple to proteins that have tyrosine kinase activity. Then we have the receptor serine or threonine kinases, which phosphorylate serine or threonine residues. Then fourth one is the histidine kinase associated receptors. Fifth is your receptor tyrosine phosphatase, which remove the phosphate groups from the tyrosine. And the last, sixth one is the receptor guanyl cyclase, which catalyze your production of CGMP, right? So right now we will discuss mainly about the CGMP, your guanyl cyclase, and your tyrosine kinase, right? So in this figure, you can see the enzyme kinase linked enzyme receptors and their transmembrane signaling by the ligand, that is your agonist. So these receptors are directly linked to the tyrosine kinase or to the guanylate cyclase, right? So when I say tyrosine kinase, these are the receptors for insulin and various other growth factors. And when I say guanylate cyclase, they are the receptors for ANP, that is your atrial natriuretic peptide. Now these both receptors that are ligand, or you can say agonist, activated transmembrane enzymes, they have this catalytic activity. Now as soon as the agonist binds to the extracellular domain of these receptors, it produces a conformational change that results in the dimerization. So dimerization is very important, which is followed by the autophosphorylation. Now this autophosphorylation of the tyrosine residue in the intracellular tyrosine binding domain. So here the autophosphorylation occurs, and then these phosphorylated tyrosine residues, they then couple with the SH2 domain of GRB2. Now this GRB2 is your growth factor receptor bound 2. So the SH2 domain of GRB2, it forms a couple with it, and it results in a series of events which leads to responses like mediating the action of a variety of growth factors or peptide mediators and that of insulin, right? There are other related receptors like cytokine receptors which control the synthesis and release of many inflammatory mediators, right? Also, there are few hormones, as I said, ANP, that is your atrial natriuretic peptide. They have also similar architecture, but their intracellular domain here is not tyrosine kinase. Here it is GC, that is your guanyl cyclase, which after synthesizing a second messenger, that is GTP to CGMP in the presence of guanyl cyclase, it will produce cellular responses, right? It will produce the cellular responses. Also, there is intrinsic enzyme linked receptors. This is basically the same thing and it is well explained here in the written form. You can see the agonist first binds, and as soon as the agonist binds to the receptor, the monomeric state leads to the formation of active dimeric state. So from monomeric state we have active dimeric state, which will activate the tyrosine protein kinase intracellular domain, okay, and then we have the autophosphorylation which takes place to the tyrosine residue, and then this autophosphorylated form, it binds with or it phosphorylates other SH2 part of the GRB, that is your growth factor receptor bound 2, and it will thus produce the cellular responses, right? Examples, we have already studied this. Insulin, example of insulin, epithelial or epidermal, only epidermal growth factor. Then we have this drug transducumab, which is an antagonist of such type receptor, which is very commonly used to treat breast cancer. Then dimerization, it also promotes internalization and degradation in lysosomes and downregulation. Similarly, we have JEC-STAT kinase binding receptors. Everything is same that we have explained earlier. The only difference is that the protein tyrosine kinase activity is not intrinsic to the receptor molecule. And when there is dimerization, it activates the intracellular domain, and the affinity to bind to the free cytosolic protein kinase JEC will increase, okay? That will lead to the phosphorylation of the tyrosine residues, which will bind to the STAT, that is your signal transducer, and activation of transcription. And this activated check then phosphorylates this dead tyrosine residues, and it will dissociate from the receptor and it will move to the nucleus. And the examples are cytokines, growth hormones, and interferons. So basically the mechanism of action of these enzyme linked receptors remains the same. Okay, so it from monomeric stage it goes to a dimeric stage. It then it then undergoes autophosphorylation, and then it dissociates and it produces the cellular responses. Okay, so this is the diagrammatic representation of what we just studied. So now moving on to the receptors for gene expression. So what is gene expression? So basically gene expression is a process by which information from a gene is used in the synthesis of functional gene product, right? And these products are often proteins, but in non-protein coding genes such as rRNA genes, or you can say tRNA genes, and the product which we get is a functional RNA, right? Now if we talk about gene expression regulation, then both of these cells contain the same genome, but they express different RNAs and different proteins, right? There are also several steps in the gene expression process that can be modulated, including transcription, then we have RNA splicing, then translation, and there is post-translational modification of protein, right? So now let's talk about the gene regulation. It includes the processes, all the processes that cells or the virus they use to turn the information in genes into the gene products, right? We have already studied this. And although a functional gene product may be an RNA or a protein, the majority of the known mechanisms regulate the protein coding genes. Now, any step of the gene's expression may be modulated, and from DNA-RNA transcription to the post-translational modification of a protein, right? So the gene regulation is very essential for eukaryotes and prokaryotes, as it increases the versatility and adaptability of an organism by allowing the cell to express protein when needed. And the first discovered example of gene regulation system was the leg operon, which was discovered by Jacques Monod, in which the protein involved in the lactose metabolism was expressed by E. coli only in the presence of lactose and absence of glucose. Furthermore, this gene regulation it drives the processes of cellular differentiation and morphogenesis, right? So it leads to the creation of different cell types in multicellular organisms, where different types of cells may possess different gene expression profile. So you can say that if we take a RNA, there is activator binding site, there is repressor binding site. There are the genes are arranged in a particular sequence. There is a promoter site. So this was the operon model, and what happens is the RNA polymerase it binds to the DNA at the promoter site, and as soon as it binds to the promoter site, transcription initiation begins, and this transcription initiation is regulated by the proteins that bind to or near the promoter area or near the promoter sequence on the DNA, right? And the repression of repressible gene, that is the negative regulation or the repressors, they bind to the operators of DNA, and which is regulated by the effectors, okay? So now if we talk about the intracellular receptors, that is cytosolic receptors, you can see from the diagram given here, you can see that this nuclear receptor family, it senses signal from the lipid soluble substance and other hormone substances like corticosteroids or sex hormones and the thyroid hormone, which influence the gene expression, right? So it has three categories of such receptors, and the main category of nuclear receptors, it belongs to the steroid hormones, and the steroid hormones are your, very good, glucocorticoid, mineralocorticoid, estrogen, progesterone, and androgens. Now their receptors are located in the cytoplasm in an inactive state, which is complexed with a HSP90, that is heat shock protein 90, and some other proteins. Okay, so glucocorticoids has a have a very high affinity towards this type of receptor, and when they cross the cell membrane, the glucocorticoid receptor then dissociates from the HSP90. It dissociates from the HSP90 and forms a homodimer, that is your GR-GR complex with glucocorticoids, and which then translocates into the cell nucleus. So this is the cell nucleus, and it translocates, and once inside the nucleus, these dimers then they transactivate, or you can say repress the genes by binding to the positive or the negative glucocorticoid responsive elements, that is GRE, glucocorticoid responsive elements. And this they do by recruiting some co-activators and co-repressors, right? And any alteration in the gene transcription leads to the change either in a desired form or in a inhibited form of gene expression. Okay. So to sum up, you can say that these intranuclear receptors are mainly have high affinity towards the steroid hormones, and they are present in inactive form, complexed with the heat shock protein, that is HSP90. And this HSP90, when they cross the cell membrane, the HSP90 dissociates and forms a homomer of GR-GR, that is glucocorticoid complex, and then it translocates into the cell nucleus, and then once in the cell nucleus, it has two options: to transactivate or to repress the genes by binding to the positive or the negative GRE, that is your glucocorticoid responsive elements. Okay. Then let's talk about the receptor regulation. In receptor regulation, so let's look into the up and down regulation of the receptors. When we talk about the upregulation of receptors, any prolonged exposure to the high concentration of agonist, whether it is administered as a drug externally or it is overproduced as a neurotransmitter endogenously, it causes reduction in the number of receptors. Okay, and if there is reduction in the number of receptors, the availability or for the activation will be less, right? And this results due to the endocytosis or internalization of the receptors from the cell surface. Also, on the other hand, the prolonged occupation of receptors by a blocker or an antagonist leads to an increase in the number of receptors with subsequent increase in the receptor sensitivity. Now this results due to externalization of the receptors out again from the inside of the cell surface, and sometimes there are other hormones which can bring this upregulation to certain receptors. For example, so to summarize, upregulation, that is increase in the number of receptors, which occurs when the activity of the receptor is lower than the usual. For example, due to long-term or prolonged administration of an antagonist. Again, for example, for upregulation, we have the administration of beta blockers which upregulate the beta adrenoceptors. Thus, if beta blockers are abruptly stopped, it can cause rebound hypertension because of the sudden stimulation of large number of beta adrenoceptors. Secondly, we have downregulation, in which downregulation it means decrease in number, that is, it is inverse of upregulation. Okay, it occurs due to repeated or long administration of an agonist. Now, along with downregulation, desensitization of the receptor to the drug may also occur, which is a physiochemical alteration in the receptor which makes it unresponsive to the drug, and it is also known as tachyphylaxis. So, two types of regulation, upregulation and downregulation. Then we have desensitization. So receptor desensitization is basically after reaching an initial high level, the response gradually diminishes over seconds or minutes. Okay, so if you continuously give a drug or give any agonist to the patient, after a certain time the response will gradually slow down or will gradually diminish. Okay, and this desensitization is usually reversible. That is one point which helps us to distinguish it from the downregulation. Okay, so there are many kinds of receptor that undergo desensitization. That is, for example, at neuromuscular junction, there is evidence that desensitization is caused by slow conformational change in the receptor, which results in the tight binding of the agonist molecule without opening of the ion channel. Similarly, there are GPCRs like beta adrenoceptors. On desensitization, they become unavailable to activate the adenyl cyclase. Okay, though they can still bind to the agonist molecule. So remember one thing, you have to know the difference between desensitization and the downregulation. Then last topic is regarding the spare receptors. Spare receptors, basically they are also known as receptor reserve. So when an agonist can produce a maximal effect without occupying all the receptors, we get a reservoir set of receptors which is known as spare receptors. Now the question is, how do cells produce a maximal effect without copying or without occupying all receptors? See, there are two answers for this. The first is you can either increase the number of receptors. So if for example, there were two receptors, right? And you give agonist or any drug, and if they occupy both the receptors, then you can say that the occupancy of the receptors for the maximal effect is 100%, right? Because there were two receptors and you gave two agonists, and they were very well occupied. Now after doubling the number of receptors, like there were two receptors earlier and now you have four receptors instead of two, but you are still giving two agonists, so they will occupy the two receptors and the two remaining will be spared, right? So you can say that there is 50% occupancy for maximum effect. The second way by which you can produce a maximal effect without occupying all receptor is by amplifying the signal from each receptor. Okay, you amplify and you get increased response. Okay. So if you want to demonstrate experimentally spare receptors, you can give the patient, or you can demonstrate it by using a irreversible antagonist, which will prevent the binding of agonist to the available receptors, and you can show that a higher concentration of agonist, maximal response can still be produced. For example, adrenaline, you can say, then we have insulin receptors, which are 90% of insulin receptors are just serving as spare receptors, right? So with this, I conclude this session. I know it's a bit inconvenient to explain all the pharmacodynamic mechanisms and signal transduction pathways properly and in depth over this media, but I hope this shall help you understand the basics at least. If you have any doubts regarding the topic, please feel free to ask. Thank you.